Frank Cole. I'm a professor of environmental science and technology. I'm a soil scientist by training and passion. And I'm also the director of the Gemstone Honors Program, and I'm very much looking forward to hearing what Gold's going to do for my life in the future.
I'm Svetlana Vukusheva. I'm head of the chemistry library. I have a long life in research, lab bench research, before I changed careers. So my area of research was biomembranes and transport across, and lipids and fatty acids. I've published a lot, and my articles are very highly cited. I hold a master's degree in chemistry and PhD in microbiology.
I'm Chris Jewell. I'm an assistant professor in bioengineering, so everything in my lab is sort of an interface of material science and immunology. So we do a lot of work on immune tolerance, like multiple sclerosis, and on cancer vaccines like melanoma and neuroblastoma. So I'm sort of working in that interdisciplinary area. I hope I never need what you guys are working on, but I hope it works in case I do.
Hi, I'm Kristen Scandal, Associate Director of the Gemstone Program, and I haven't taken biology since before you were born, so I'm on the lay side of the audience.
I'm Ben Woodard. I'm the Director of the Biotech Research and Education Program here at the University of Maryland, and I am the mentor of Team Gold. Previous mentor of Team Silver. This is a slight continuation of what Silver was able to accomplish in their 3 years, and we hope that this group this year, next 2 years, can really grow upon that research and maybe get something that's some individual IP or something that can be patented or created, some type of valuable product.
Classic first-hand example of the incremental pace of research.
Yeah, exactly.
One step leads to another step that came from a previous step.
I think Team Platinum might actually get something that's
And then Team Carbon Fiber. Okay, Team Gold, it's all up to you. Go.
Hello and good afternoon, everyone. My name is Dimitri. I am joined by Parmita and Margot, and we are here representing Team Gold to discuss the problem of antibiotic resistance and how our research plans to eliminate this problem. Antibiotics were once things of absolute miracles and still are to a certain extent. Lethal infections could be treated with ease with antibiotics such as penicillin, which was once regarded as a cure-all, but today has lost most of its effectiveness. This is due to the increased usage and distribution of antibiotics. Bacteria has evolved and grown resistant to these miracle workers, which leaves the body once again open to attack. This is a problem that affects every single human being. There's not one person in this room who wants to contract a MRSA infection with no way to combat it. Hope is not lost, however, because there is a way to develop a bactericide that is not susceptible to the development of resistance. Now, one method of killing bacteria that doesn't lead to resistance is to manipulate the natural processes of the cell. Specifically, we will target bacteria's iron uptake pathway. We plan on so conveniently, these bacteria naturally produce siderophores, which are these molecules that are secreted by bacteria to scavenge for iron in the nearby environment and then bring it back to the cell. And this is a vital mechanism because iron is necessary for essential life processes. What we plan to do is to form a complex with gallium ions and the siderophore desaferoxamine to form a GADFO complex that will go inside the cytoplasm of these bacteria cells. This is an important modification because unlike iron, gallium is toxic to bacteria. Now, many bacteria have adapted to accept foreign siderophores in order to maximize their iron intake levels. So this means that one siderophore can enter the membrane of many different types of bacteria. And this is the adaptation that we plan to exploit with our GADFO complex. A good analogy for our method of attack is the story of the Trojan horse. In the metaphor, the Trojan horse is the siderophore, the soldiers are the toxic gallium ions, and the city's wall is the bacterial membrane. And just as the city of Troy was tricked into accepting a Trojan horse that ultimately led to their downfall, we are creating a Trojan horse with the GADFO complex that will essentially be tricking the cell into accepting toxic gallium ions instead of iron ions into the cytoplasm. Now, previous research has shown that the GADFO complex is successful in inhibiting the growth of several species of bacteria in an iron-poor environment. We seek to expand upon this research by testing the complex in different media with different concentrations of iron against a broad spectrum of bacteria, over different lengths of time and in growth conditions that are naturally found in the environment. Our criteria for selecting which bacteria we will culture include the gram type, the biosafety level, its clinical relevance, and its ability to form biofilms.
Can you say something a little bit about, so the first point, it's been used previously, like, is that in cell culture, in people, or clinical trials?
That's against the previous bacteria that's listed there.
But is that, is it like an actual treatment currently, or is it just, it's been like in cell culture shown?
It's just been in cell culture.
Okay.
Has been tested in any other areas as of yet. And that's what we seek to expand upon. And we have added the future bacteria. This is what we'll be adding to the list of what we'll be tested against. And this will be selected based on these levels of description.
Why you pause there? So the biosafety level, is that dictated by the types of bacteria that you can comfortably or safely manage in the laboratory, or is there some other reason that's in the list?
So first, the previous bacteria are all biosafety level 1, but we want to adapt to biosafety level 2 and get the required training because those bacteria are mostly found in hospital settings, such as catheters, so we want to attack those bacteria and see how the Siderophore affects them.
Okay, do you have access to a BSL-2 lab?
Yes.
Okay.
Why wasn't Pseudomonas aeruginosa included on your list? It is very important.
There's been extensive research on that species already. There's actually like a paper published about that. It's been GADFO has been found to be effective against that strain in planktonic growth and biofilm growth. So we figured that since there's already been so much knowledge on it, we can like focus our research on other species.
I don't think the wind's gonna blow us out of here.
Beyond testing the GADFO complex with this bacteria, we will also be testing the effectiveness of the complex in mammalian cell culture. So based on these aims, our team has developed 4 research questions that will be the basis of our research over the next year and a half.
So the first step of our research question seeks to determine the minimum inhibitory concentration, which is a measurement of toxicity of our GADFO complex against a broad spectrum of bacteria. Now we would also like to test its effectiveness over a long-term and short-term period, which will be 1 month and 2 weeks respectively. Now, in order to generate the GADFO, we will have to synthesize it ourselves. In order to determine if the complex has successfully formed, we will use a CAS assay. Once we have determined that the complex has been successfully formed, we will grow bacterial cultures that we intend to test on in shake flasks to mid-log phase, and then administer one of the following 3 treatments. The first will be a buffer control. The second will be a free gallium ion treatment. Now, the free gallium ion treatment is necessary because we want to determine whether the complex stays within the cell or is released back into the environment. The free gallium ion treatment will serve as a point of comparison for the DFO delivery mechanism. And the third will be the GADFO treatment in varying conditions.
So the CAS assay, can you say something about how that works?
So it's going to be, we're observing the color change in the reaction, and so it's going to measure the mobility of the iron ions, and it's supposed to change color when there's a difference in concentration for that. So we'll be using spectrophotometry to measure that and see if the siderophores have successfully are working culture in there and doing what they need to be doing.
So you add some kind of reagent,
Yes.
and it interacts with complex iron differently than
And
free
it's,
iron?
and it's supposed to exhibit a different color change.
We will determine growth rates with both microplate fermentation and CFU analysis. Microplate fermentation records information about the optical density, and CFU analysis records information about the viability of cells. Using the data from both of these analyses, we will extrapolate a growth rate after overnight incubation and determine the MIC of our GADFO against a bacterial cultures. The last part of our first research question seeks to determine the effectiveness of this complex over a long-term and short-term period. To do this, we will reinfect all cultures 1 month and 2 weeks after initial treatment to determine its efficacy. Now, if GADFO proves to be effective in iron-poor environments, we will continue to test the complex in iron-rich environments. Now, this is important because mammalian cells are rich in iron, and to want to create a product that affects mammalian cells, we have to be sure of its efficacy in iron-rich environments. Now, this is important because the complex depends on bacterial cells scavenging for siderophores. So if an iron-rich environment hinders this, we would have to test it to determine how so. Now, in our initial studies of our first research question will be replicated simultaneously in an iron-rich environment with the same 3 treatment groups of buffer, free gallium ion, and the GADFO treatment in varying conditions. To create iron-rich environments, we will be using the standard biopharmaceutical media that already has iron. If the MIC in the iron-poor environment isn't altered in the iron-rich environment, we will move on to investigate the effects of the complex in a biofilm setting. Now, biofilms are aggregations of bacterial cultures that exhibit much higher levels of antibiotic resistance than any normal culture of bacteria. They are relevant in clinical infections in medical implants and damaged tissues. They're also very difficult to treat with traditional antibiotics, so a new treatment would be very effective against biofilms. Now, biofilms cannot be grown in shake flasks because they require a substrate to attach to. They will be grown on a semi-permeable membrane sitting on top of an agar plate. Treatments will be administered to the agar medium with the same 3 treatment groups as before. No treatment, free gallium ion, and the GADFO in varying conditions. Now, some advantages of this are that this minimizes the immigration and emigration of cells, so all changes in cell count can be attributed to cell death. Some disadvantages, though, are that growth rate cannot be measured using the aliquots of the culture. At the end of the incubation period, the colony will be centrifuged to the membrane to remove the bacteria from the membrane. The resulting diluted cells will be plated onto colony formation units, and then the CFU analysis will be used to extrapolate a growth rate. Now, these 2 questions expand upon previous research by testing the GADFO treatment as a potential alternative to pen strep. Pen strep is the industry standard in mammalian cell culture. So after we have confirmed the efficacy of our treatment against free suspension and biofilm bacteria, our next step will be to determine the efficacy of this of our treatment against mammalian cells. Now, testing on mammalian cells is important because almost all applications of our treatment involve an environment where this complex would come into contact with mammalian cells. If the treatment did harm mammalian cells, however, we would nonetheless be able to apply our research. However, we would be more limited in potential applications. Now, once the MIC is established, we would need to verify that this concentration is not harmful to human cells. We would do this by adding the GADFO treatment to a mammalian cell culture alone to determine if it's harmful. The dosage that we would administer using the MICs found in our first 2 research questions would be used to administer the GADFO. We will culture HEK cells, and then we will infect a mammalian cell culture with bacteria and attempt to treat that infection with the GADFO as a simulation of a real-life application. If our original dosage either fails to kill bacteria with the HEK cells present, or kills both the bacteria and the HEK cells present, we would adjust the concentration. We would also administer a control treatment, pen strep, to better understand the viability of our treatment against the industry standard. Even if the GADFO proves to be harmful against the HEK cells, we would still carry on to see how we may apply GADFO to applications that don't directly involve humans.
So this is a visual summary of our methods discussed by Parmita in the form of a 3-year plan of how our team is going to be working towards the completion of our research. We will first be retesting the strains included in previous research, And then we'll be testing additional strains as well, as we specified earlier in the presentation. So this part of the experimentation will all be taking place from approximately March through May of 2016. Upon returning in the fall of 2016, we will be performing our testing on bacteria in the form of biofilms and on the mammalian cell cultures. The results from these tests are really critical because they give us a lot of information about the clinical applicability and the commercial potential of the GADFO treatment. In addition, as we begin writing our thesis, we expect to perform further testing based on the results we obtain from the methods outlined in this presentation. So we do anticipate that we may come across some possible challenges throughout our project. We've identified some of these potential challenges to be effectively scheduling our times in the lab for experimentation, learning how to properly use the colony biofilm assay, successfully growing mammalian cell cultures, avoiding contamination of our cultures, and dealing with some unexpected results. For example, if we do see a lack of treatment effectiveness in the iron-rich environment. So for our lab schedule, we have the ability to utilize all 10 members if necessary. Each person in part of the laboratory sub-team will be scheduled to perform a particular part of the experiment at a particular time. For collecting our data on biofilms, we know that it will take some time to learn and adapt to the colony biofilm assay technique, so we have allotted enough time in our 3-year plan to practice those techniques in order to ensure that they are performed efficiently. To ensure successful mammalian cell culturing, we will have at least one team member spend the summer gaining competency in mammalian cell culture growth. That person will then make up part of the mammalian cell culture sub-team performing tests on HEK cells. Contamination is a common concern among researchers in the laboratory, so we aim to just minimize this concern as much as we possibly can. Instead of all team members working in the lab simultaneously, we will utilize small sub-teams in order to minimize potential exposure to contaminants. In addition, our team members have been trained in proper aseptic technique, which will be strictly followed throughout all of our experiments. If we find the proposed treatment to be ineffective in iron-rich environments during the methodology for research question 2, the following research questions and methods will be modified accordingly, as we touched on previously in the presentation. We're completing our research with real-world applications and potential product development in mind. Overuse of antibiotics is a huge issue in the fields of agriculture and biopharmaceuticals. So GADFO could offer an alternative that is more favorable than antibiotics and thus offers a lot of commercial value. In addition, there's a possibility of creating everyday commercial products, such as AstroTurf, nail polish, cough drops, and a variety of different items, all with antimicrobial properties using the GADFO treatment. Earlier in the presentation, we discussed antibiotics and the implications of antibiotics, particularly when it comes to human health. We expect the GADFO treatment to offer a novel and effective method of treating bacterial infections and avoiding the evolutionary pressures that lead to antibiotic resistance. So thank you so much for listening, and we'd like to open the floor for any more questions you may have.
Great, thank you.
I have a question about how do you plan to manage results and data, because you're working on it as a team. Let's say one subgroup has some results. How are you going to coordinate and enter the data and manage the data?
Well, that's one thing I think that Google Drive will be very helpful for. They have a feature where you can sync documents so that everyone can access them from their own computers at any time. So we've been utilizing that a lot to create our presentations, to write our papers. So with entering data, we think we'll be using that too.
Maybe you could think about using an electronic laboratory notebook rather than research technology. And I can talk to you about it. We're experimenting with it in some chemistry courses right now.
Okay.
Joe, you care to ask some questions?
So back to kind of the question I asked earlier about what's been done. So if it sounds like there's some promising in vitro like data in cell culture for this approach. What do you think the hurdles are? Like, why hasn't it been, why isn't it everywhere in AstroTurf and everything already? Like, are there reasons that this technology hasn't been developed yet, or just wanted to get your thoughts on that.
I think it's still a novel approach that not many people know about the bacteria, the GADFO's effectiveness, and I think people are still trying to expand on this research. We are just hoping that our rate is faster than other people's rate and we can come to a commercial product.
Yeah, okay.
I mean, it wasn't that long ago before the GADFO complex was like, synthesized and patented, so that's just been a recent development too, which was what we'll be using.
Like the paper that I mentioned earlier on Pseudomonas aeruginosa, that was published I think around like 2009, and I think like if you're going to develop something that's going to be used for humans, that has to go through a lot of like preliminary trials to make sure that it's effective, and then you have to like test on animals and then go through FDA approval. So I mean, maybe there is something that's being worked on somewhere else by another scientist, but I guess like research hasn't come out about that yet.
Yeah, okay. And when you actually make the complex, let's say you take me into the lab, hopefully I don't destroy anything, but how do you actually make these?
Well, we'll be purchasing the GADFO siderophores.
Oh, so what's the CAS assay? That's just to...
We're purchasing the desferoxamine. We're using the CAS assay to create the complex, and then with the color change that will indicate if the complex has successfully binded to each other.
Yeah, so it's gallium. It's a solution with free gallium ions, and we add in the DFO, and then like the DFO will sequester the gallium, and then the CAS assay basically is just a color change of an indication of free gallium ions in the solution. So if there's less gallium ions, the color will change.
So when you actually make it, it's just a mixing together.
Yeah. It's a chelation assay, the CAS.
Okay.
Yeah.
So anything that chelates, the color changes based upon the removal of the ion from
And
the...
it'll confirm whether it occurred or not.
Yeah. Okay. And for your... So what was the difference in question 2 and question 3?
So question 2 was trying to test the GADFO in an iron-rich environment, and then question 3 was trying to test the effectiveness in a biofilm environment.
So, okay, so, because in both you'll have the bacteria in there, right?
Yes.
Okay,
Yes.
and then is it just whether it's biofilm or in the free-floating form?
Yes.
Okay.
Can I follow up on that?
Yes,
It's
sure.
a very similar question. Now, the biofilm, is that, correct me if I'm wrong, the importance of that is that's when you have the bad germ on an inert object like a catheter or some other piece of plastic or glass, whatever. So that's, just want to ask the question, is that by definition a low iron environment? Because it's not with the whole slurry of cells?
No, I believe there would be iron present in that environment, and that's why we're not looking at the biofilm environment without iron, because that's not really something that you would see in everyday life. You would, if you have a biofilm environment, you would see iron as well.
So part of the physiology of a biofilm, if that's the right frame, would be a high iron environment.
That's why we're doing the iron-rich environment before the biofilm, to ensure that the complex is actually effective in an iron-rich environment.
All right, makes sense, thanks. Sorry.
So another follow-up on the biofilm. So in this research area, one of the important things that impacts biofilm formation and resistance and things is whether it's a flow setting or whether it's a static. So like in cell culture, if you just have immediate static and if it's in blood or something, or if it's in fluid flow, then that can, is there anything like either you'll look into that, like static versus dynamic, or if not, what kind of things do you think you might have to be aware of or limitations?
Yeah, that is something that we can test with static versus flow. Right now, we only have a procedure for static, and obviously, it's a new procedure for all, it's a new protocol for all of us. And given the time constraints of 2 years to test everything that we've laid out, we don't think there'll be time to necessarily test static, and that's an area that can be built upon later.
Okay, yeah. So you prioritize that. then what do you think what does that mean in terms of your conclusions or how you can apply your findings, or does it not matter, or is it like, is it relevant to 1% of cases, or?
I think it definitely matters, especially if this were to be administered as medication or something like that in order to treat an infection within the body. But first, we're taking the steps to just see if it harms mammalian cells. So I think after that kind of preliminary research, like we said before, we would expand upon that later.
Feel free to take notes and sit down and capture this information, because you may not have this opportunity again, so.
Yeah, and I'm typing up I'm not paying attention. I'm typing up my stuff.
Okay.
So I'll send it to you as well.
I have a question. Is there a way to measure the iron concentration in this biofilm? Because you said it's iron-rich environment, but how rich? How much that could affect the interaction with gallium?
So we'll be purchasing our own media to grow biofilms in, and normally when you do buy media, they have specifications for like what the content of it is. So that's basically what we're going to use to measure the content of iron in our media.
But is there a reference of what a typical biofilm iron concentration might be, and what form that is? Is it free? Is it complex? Or I'm still hung up on this biofilm thing.
So you're assuming that the concentration in the biofilm for iron will be the same as in the medium?
Not necessarily the same, but they will both be rich in iron, as opposed to our first research question, which it's very iron poor. So for our first research question, we would be specifically purchasing iron-free disposable consumables, and can I have it for just 2 seconds? Sorry.
Oh, while they're doing that, I think I'd like to address maybe the point of confusion here is that the complex isn't formed inside the cell. It's actually formed outside of the cell. We're making it separately and adding it to the media, and the mechanism is that the potentially toxic compound is brought into the cell where it'll kill the bacteria. So, I think what's more important is the concentration of the compound outside of the cell, and that's where it'll be more important. Does that?
So to address our first research question, we will be specifically, like I said, purchasing iron-free disposable consumables to ensure that the media that we're testing it in is very poor in iron. If that proves to be effective, then we will move on to iron-rich environments. So our first research question is completely free of iron.
So when you do this question, or even in like question 2 or 3, if you find that, you address this a little bit, but if you find you don't have any effect, how will that impact your research plan?
So that's a good question. If we find that there is no effect on the iron poor environment, then we will have to adjust our third and fourth research questions accordingly, because we would not be able to move on to biofilms without having good results for our second research question.
Do you have any ideas, it'll depend what the results are a little bit, but what kind of things might you adjust or change or modify?
So if it proved to be not effective in iron-rich environments, then we would have to we would not be able to use it in a mammalian cell setting, because mammalian cells are rich in iron. We would then move on to potentially creating agricultural uses of the product. We would see what the research would tell us.
Okay.
So you have a potential fork in the road coming up. So if it proves to be effective in a low iron environment, you can move on to, let's see what it does in a system high iron environment. If it's ineffective in a low iron environment, then you have to figure out what you're going to do next. I think it's a great setup. Okay, your threshold for effective and ineffective, it may have been in the paper, I read it sometimes, I don't recall. Is that a moving threshold, or is there an established efficacy, non-efficacious cutoff? Or how are you going to decide which fork you're going to take? When's it effective or when's it not effective?
So that would be our minimum inhibitory concentration. So I guess that would be more of a threshold kind of thing, not like a specific number that not a specific thing that we're looking for. I guess we would just have to see what the results of our first and second research questions suggest, and then using those two combined results, we would then adjust our third and fourth research questions accordingly.
So you're gonna have an array of responses over concentrations to find that MIC,
Yes.
right?
Yes.
And if it's a linear relationship, if it's a straight line, how we decide what's high enough? Or if it's a classic plateau or the other direction where there's a nice break point to the data, you can say, well, that's where it is. So have you gone into it with an idea of how you're going to identify that, or it's just going to be from data analysis once you get your first round of data?
The MIC. So this was addressed here. So we would use microplate fermentation and CFU analysis to determine that MIC after overnight incubation. Once the MIC is determined, then we would move on to its long-term and short-term.
And the MIC is only for one concentration. Like the MIC is the concentration of the compound that will inhibit growth after overnight incubation. So this is where our control is going to be important. We're going to do an ANOVA statistical test of variance. So to compare the MIC for one bacteria for the treatment to the control, which is the buffer. So if it differs by a significant amount, then we'll treat that as a measure of efficacy.
So it's separation of the means of the populations of data.
Yeah.
Okay, thank you.
So if you, kind of on the same line of question here, if you look at the other papers where they've done like different strains, does that tell you anything about like the iron levels or the impact of?
Yeah. Actually, can I see it? We have a chart there have been other experiments done on other strains of bacteria on other papers, and they found some inhibitory concentrations preliminary, and here you'll see this is the list of microorganisms, the treatment compound, and then the medium that the experiment was performed in, and there are available inhibitory concentrations on these, and I'm sorry, what was your question?
I was wondering, so if like this chart actually is really good, do you know, does it provide any information about iron concentrations?
Yeah, so that would be found in the description for the medium, because so they listed out the medium that they use, and if you wanted to find out the iron concentration, you have to go back and check with the manufacturer's description.
So if you hypothesize based on that, like is it likely that it's going to work in low or rich iron, or? Because if you have some data for that from previous literature, then when someone asks, you can be like, yeah, we're proposing this, but these other two groups did this, and it worked at low concentrations, or it worked in an iron rich, or you have some evidence to support your idea to test other strains.
Yeah, so that's actually a problem with the previous experimentation, is because you'll see that some of these media were like undefined, so there really isn't that much good data on whether or not it works in iron rich versus iron poor. I think most of the papers that we've read have been done in iron poor environments, so this is where like our building upon previous research comes in, like we want to contribute more.
If you take that data, or if there's more data to it, and you do a simple little plot of the iron concentration in the substrate media, whatever it was, versus the inhibitory concentration, you might see a pattern that you can kind of use to target your concentrations with.
Yeah.
Now, so the way this mechanism works, it's like you've got a bacteria, it shoots out the siderophores, they sequester something, and they go take it back in, right? Like iron, and it uses, okay. So does that happen in mammalian cells at all? This might be like a really or
That doesn't
Okay.
happen in
Cells
mammalian cells.
Yeah.
We're using mammalian cells
no, I understand. I just wanted to make sure.
Yeah,
No,
but
yeah,
mammalian
no.
cells do
Okay.
not.
So
secrete.
then back to the bacterial cells, is every strain, like is the binding and uptake of the siderophore complex after it's bound its target the same? Like are they all the same pathways by strain?
Yeah,
Yes.
so that's where the siderophore piracy that was mentioned at the beginning of the presentation comes into play. Like siderophores all produce like their own special type, but one of the things that they do is they have receptors for many different siderophores from other species to like maximize their iron intake. So that's why we think it'll be effective against a broad strain of bacteria.
So I'm just thinking about like when you start doing some of these studies, your endpoint, your readouts right now are like, did it kill the bacteria, basically, right?
Yeah.
But if you can think about, it might not kill bacteria because it didn't get in, because it bound but couldn't be internalized, or because it didn't bind at all, right? I mean, there are different reasons it might not kill the bacteria. I'm just wondering, like, if you find that it's not working on different strains, if there's some kind of mechanistic studies you could look at to see, like, is there a certain step where something's happening? I mean,
Yeah, that's definitely possible. You could do like a blast analysis for the receptor to see like which bacteria have receptors that are similar to that. Yeah, something we can look into that Silver actually did, but we haven't really thought
Yeah.
of.
And I think
But
we
I
can
think
look
it'd be
at
very
trends between gram-positive versus gram-negative, or trends between, these bacteria have this certain receptor for whatever type of siderophore, and see if that has
So
any effect on it.
let's say, you bind the gallium, no iron's getting in, the gallium's getting in. What would the cell dies, right? But what would you expect, like, why does it die?
Why the
bacteria.
mammalian cell? Or?
No, the bacteria.
Oh, because bacteria need to reduce iron from iron like 3+ to like iron 2+ or something, but gallium you can't do that in the cell, and then it just accumulates and it's toxic.
Toxic to it.
So the part with the iron, if it can't reduce the iron, like what is, I
You
mean,
can't reduce the gallium.
oh, it can't reduce the gallium, but I mean, it wants to reduce the iron, right?
Yes.
So if it can't do that, like what's the effect? Why does it die?
There are metabolic pathways that
Uh-huh.
utilize iron. I don't know specifically
Basically,
with
be
Patrick.
starving the bacteria so it won't get the iron it needs to live.
Because I mean, I think there are a lot of pathways involved in that you could actually measure, and that would actually be nice because you could like try and figure. I mean, if you wanted to like patent something, mechanism of action can be important.
Right.
Like so, even if it died, it's probably a very good chance these iron pathways and the metabolism's messed up. But even just if you see it die, then you could just measure that. There are different. There's MTTs in easy metabolic assay. Usually it's on mammalian cells, but I think there's something similar for bacteria. You could find genes involved in the pathways that you would expect to change if the bacteria is dying, and you could think about there are lots of different ways to measure gene expression, but because that could like amplify your results, but also if you don't get the result you want, it might help you tease out why.
Right.
So I would just keep.
Yeah.
I wrote a note here, but I would just keep in mind like some of there are mechanistic things that you could do, which I don't know what lab you're doing it in or whatever, but if they're doing bacterial work, they probably already have some kinds of different techniques that could be useful. So.
Something we
Yes,
can
more
do.
questions?
No, not right now.
Okay. Chris, you have some questions you want to interject? Anyone interject questions?
No, I'm good. I'll let Stare
Okay.
research. I'm just here to push them in the right direction.
Shifting focus to how a team and how you'll function with your subgroups, how will you hold each other accountable and communicate so that you can accomplish your goals?
So one thing that we have done is we've set a kind of penalty, I guess, for not coming to meetings or not telling us ahead of time. If you have a reason, then that's acceptable. But if you don't come to meetings, you have to provide snacks for the next meeting. So that's something that's less severe than like you don't get to participate at all. It's just an incentive to come to all the meetings.
So how will you translate that to when you're in the lab? Because if you are on a sub-team and someone's growing the media and they set it up and then someone else has to come in and do the next step, but you haven't communicated something, or someone doesn't come to the lab when they say they will, so now your whole timeline is thrown off. How will you make sure that when you're in the lab, you're particularly holding each other accountable?
So one thing that we would do to ensure that is ahead of time we would use when-to meets to set up when people are available to use the lab, and we would make sure that that person knows that they have to be there. And I'm sure everyone on the team knows that if you don't show up to one lab time, then that could compromise all of the research we have done up to that point. And I'm confident that our team will come to the lab time scheduled.
And we're actually this week getting into more specifics of who will be making up that laboratory team, and I think we'll definitely get into more specifics of the consequences for that as well, because we're just starting our actual hands-on research in the laboratory.
And consequences isn't necessarily the biggest part. The biggest part is to make sure you have quality data from person to person, group to group in the lab. So like communication continuity, that reliability, that the step A is going to lead to step B. Yeah, I think bringing snacks is a great consequence. But,
Don't bring it to the lab.
but the idea of finding a way to make sure that the data generation is solid and continuous from person to person.
Yeah, and we also will be discussing our own strengths and weaknesses when we're determining who's making up each sub-team, and the people who have strengths in that certain area will then get further training with Ben and on those different techniques, and then
Excellent.
they'll be a little bit more of experts in that part of the experiment.
And do you have someone who will be doing the mammalian cell culturing this summer? Okay.
Okay. Done. A couple more kind of follow-up questions. You have, the budget you presented in your proposal was, what, $1,500 or something like that? Pretty modest. I mean, I read your proposal, which I think is great. It's got a lot of ambitious goals, 4 or 5 different aims, and can you pull that off for $1,500?
We have a lot of the materials and equipment that we're utilizing donated or available on campus. We were lucky enough to be continuing Silver's research, so we do have a lot of their leftover materials.
Can I stick with her?
Yeah.
We actually have a slide.
Yeah. That's what. Wrong computer.
Oh, there we go. This is our budget. This is the initial.
Simplified.
This is the simplified, and then this is the
Okay, yeah, so 13, yeah. So it's a very doable budget over 3 years, which is good to know from a lot of different reasons. But as long as you think you're not undershooting the cost. You don't want to end up being, oh my God, we need to have twice as much money when it gets critical time. So that's something you should be able to achieve. And you started searching for outside sources of funds?
Yeah, we're applying to the library award, but in addition to that, we actually are starting with around that much money already. So if we are able to get any more funds, that would be great.
Yeah, I would try and get them.
Yeah,
Yeah.
we're trying.
No matter what, almost. You want more for
Yeah.
research, but especially biologically related research. It can be
Yeah.
expensive.
Yeah, if you decide to use an electronic laboratory notebook, it costs small money, of course, you can have it as a group, but maybe, I don't know what they can use funds for that. I don't remember how much it is, maybe it is $40 per person per year or something like that.
Okay. That's something, if you want to go that route, you have to build that into your budget.
And think about 2 conferences, teams have gone
And
to
travel.
biomedical, BMES, I forget what that stands for, and
But-
Team Silver went to somewhere. in
Microbial
San-
control, RAMC.
Yeah,
Recent
and
Innovation.
BMES definitely has, it's a fairly new program actually. It's called Undergraduate Research and Design.
What's that one?
Biomedical Engineering Society. It's in October every year, and the deadline for abstracts and for undergraduate research and design awards, and I think they're like a couple $1,000s, is I think like April or May.
So if the deadline's April, say April 15th, that's a deadline for other things in this world. The deadline's April 15th, then when's your proposal have to be completed and start circulating through the university approval process?
the deadline for.
If the deadline for the proposal to be submitted by the agency is April 15th,
Okay.
when's your work need to be finished here, and so you get the proposals routed and through and out the door?
Yeah.
I believe it's a month ahead of time.
Yeah, March 15th.
Yes.
So, plan a month early.
I mean, it could be in May, and it could also be something like next year, or, I mean,
Yeah.
there's a lot of, there's Northeast Bioengineering Conference.
So we started working on an HHMI, but then realized it stopped this year.
Yeah.
Yeah.
So they've got a big packet together, ready to go. Just
Oh,
need to
good.
find somewhere to send it out.
So if everything goes well, like what are the outputs and what's sort of the timeline? I mean, you have a timeline, but I just want to hear your thoughts on it.
So I think if everything goes according to plan, if our first, second, third, and fourth research questions, we get the data that we want, hopefully we would be able to create one of the products that Margot was talking about. It would either be in biopharmaceuticals, agriculture, or everyday use like fingernail polish and cough drops and everything.
Yeah, I think
Go.
one thing to think about is we kind of are casting a wide net. Is it effective against mammalian, or effective in cultures with mammalian cells? That's an application in the pharmaceutical industry with culturing mammalian cells. If it's effective against biofilms are contagious everywhere. So, I mean, there are a range of applications where it can work. So, depending on the results we get, we want to tailor it to where the product would be most effective.
I guess I meant, so I mean, definitely we have whichever your favorite product is, preventing this is good. But so there's a lot of steps to go from we're going to start some studies to it's in being used by people. So I meant is there a certain stage where okay, if we get this data, we're going to have a paper describing these results, or if we get to this stage, we're going to file a disclosure with the university. you know, for the initial stages of patent protection. Or like when we get to this stage, we can go to a conference, you know, like more like sort of milestones that might get you towards this goal but are more concrete and achievable in, you know, sort of a planned way.
Yeah, so we actually think there's a possibility for a paper after our studies in iron-rich environments, because that's something that hasn't been tested before, and we think that our findings there could be significant in the field.
Okay.
Yeah.
So when you start doing your experiments, you know, almost immediately, this is my advice, but almost immediately you want to like map out what would a small paper or a big paper or whatever paper look like. Like figure 1 would be, you know, you just think about like the theme, like figure 1, this is on low iron, what happens? Or making the complexes, we made them, that's figure 1, right? And whatever data, figure 2, we need something on low iron. Figure 3, we did that again, but now we did it in 4 different strains, right? I mean, you just kind of, when you start getting some data, then start thinking, what would we need to tell the story? Because that'll help, I think, keep you focused on, like, you know, tangible productivity, which is good for your project, but also good for all of your CVs, you know, if you can get a paper or a patent or something, so.
Love.
I think after research question 2, if we get the answers that we want, that has the potential for a small paper. And then if we get the answers we want to research question 3, which is the biofilms, that has the potential for a bigger paper. And then if we get the answers that we want to research question 4, then hopefully we will be able to patent a product and have our names on it.
Yeah.
Very sweet. Good ambitious plan. I have one question I want to ask you from, this would be a question I suspect you might hear from a more lay audience, who, if you try to pitch this for funding or whatever different arena. So if I'm a bacterial cell and I'm living in a high iron environment, why would I waste the energy to send out a siderophore to go find iron? I should have enough of it just around me, right? I'm swimming in it.
Well, the bacterial cell still needs to have a mechanism to bring that iron in. The iron can't just get into the bacterial cell. So the iron sends out these siderophores to scavenge for iron. If we are able to manipulate that pathway with the GADFO, it would take in the gallium, and then the gallium would, in result, be the bacterial cell's downfall.
Thank you. So the bacterial cell, I'm not responding to my environment to decide to send out the siderophore or not. I'm doing it anyway.
So if the siderophore is designed not to collect iron, the bacteria cell's not going to get iron.
Good. Good. Well done. Any other follow-up questions from anyone?
I have a couple. Just, I mean, overall, I thought it was really good. I liked the presentation or the paper. I thought it was nice and clear. Kind of what you just mentioned. I think having a schematic or something would be really useful. Like of, you know, in your document. I don't know if there's a certain place you're supposed to put them in the document, like at the end or the beginning or in the text, but just that is kind of like, oh, here's a bacteria, you know, and it sends these out and it grabs the iron and brings it back. All of a sudden, if this is now gallium, it brings it, you know, and it's like your Trojan horse thing, right? It kills the cell. So I think that would make it really clear to someone who isn't necessarily going to study it or, you know, doesn't have that kind of training. And then there are some minor things I marked, like one place I think you have like, we've got 4 questions, but then you list 5, and a few minor things like that I marked. But overall, I thought it was really good.
Yeah, Prozel is in good shape, and this is the point in time where you are launching onto your research path. This is the point where we say, good job. You laid out a concise and clear path for how you're going to move forward. I think it's very ambitious. Don't lose sight of the fact that if you get to those forks in the road we talked about and something doesn't work out on one fork, there's always another question to ask and change it as you go. And that those successes about when we're going to write papers, when do they come? I heard, I think he said there's a small paper than a larger paper. There's no such thing as a small paper. They're all wonderful achievements. Okay, so each achievement is wonderful. Don't downgrade something because it's not as grandiose as you originally thought it might be.
Yeah.
Great accomplishments.
And small and big, they don't have to refer to like quality or achievement. It can refer to scope, like, you know, they're different. But yeah, I agree. And once you have your ideas of the data coming in, like, how can you like convert that to tangible productivity? That's, I think, that's really important.
I think that you did a great job. The text is excellent. I think it is very well written, and your presentation was very good. I think after another option, in addition to an article, at least you should put it in DRUM, the repository of the university, as other theses have been put. But I think you did a great job. Even it is very professionally written, the proposal. So.
Yeah.
Do make sure you do all of your proper lab training, and that you have them all
I
through
think
the.
they're almost all
Mhm.
done.
All done,
now.
okay.
All
Yeah,
done. We did the basic last semester, but you have others to do,
Yeah,
especially
BSL
those BSL
too.
two.
Yeah, the BSL
Yeah.
to make sure some of those can be, you know, you can work with them safely, but they can be dangerous.
And you want to make sure that you have the supplemental online trainings to go with the in-person one thing.
Yeah, and once you start doing experiments, just don't be afraid to like change course. You know, don't, if it's not working and you've done the right experiments and test the right things, cut it and change it. And just don't be afraid to do that, even though you might have invested, you know, time and sweat and blood and tears in it. That's gonna be better.
All
in the
those
long run.
things.
Yeah.
Hopefully not too much blood.
Yeah.
For your final thesis, because it is published in Drum, even if you have no other papers, any images that you use, either you should create them and cite yourselves, or make sure that you reprint them with permission, and there's various ways to do that. So any images that you use that you don't create do need to have the permissions.
Just one last thing, because you have to write something after all. It is just the beginning, but start thinking about how you are going to present it, even when you are doing the experiments. How does this result going to look in some final article? So it is very important to think about it all the time, not just at the very last moment. So we have these results, but how do we interpret them? Where do we put them? So I think it's
Do you know who George Whitesides is?
No.
Okay. He's a famous, let's just call him scientist because he's done a lot of things at Harvard, but he has a very famous paper that's been cited 10,000, 20,000 times. And it's basically, it's only a couple, it's like originally typed on a typewriter. It's only a few pages, but it basically is like how to write a paper or how to approach science, I think is something. And he says, some people disagree with this, but he says, before you do anything, you outline the paper. So, you know, whether you think it's that extreme or not, but thinking about it like as you go is important. And I just thought the Catalyst, do you know the Catalyst?
Oh, yeah.
So that could be a good, that's like a, is it run by bioengineering department?
Yeah, it's
or-
bioengineering.
So it's like a student journal, but I just saw I don't know if it's the first issue or one of the first
The
issues,
first
it looks pretty
print
good.
issue, I think
Yeah,
they did.
so I think that could be a nice, like, you know, have that and you list that just like anything else. I think there's some peer review to it. And then, you know, if there's other external things, definitely pursue those too.
Well, good job, Cole.
Yeah.
Yes.